Morning to all of you this morning. It's rainy here, but anyway, getting a little bit better. I feel a little like Fred Rogers this morning wearing this red cardigan. Anyway, it is a good morning. That is until I listened to the BBC News and I heard the some commentary on the COP26 UN summit thing, and I'll just give you a couple of quotes. One was and these quotes came from two climatologists. What we do now will be felt for centuries to come. And looking at past records, I have never seen anything encouraging. Now that was a young lady, a climatologist who's working in the Antarctic and has core samples going back thousands and thousands of years, and she just hasn't seen anything like this. So really quite extraordinary. And another said that even if we stopped all CO2 emissions now, never mind keeping it within the 1.2 degrees centigrade, Greenland will continue to melt. Well, that's certainly what my daughter's experience is up in Alaska. And she took me on a walking tour electronically yesterday. Wow, what a change, particularly when she provided the footnotes of what it had been like 2 years before, 5 years before, and earlier than that. So it's a little grim. So I hope that they steel themselves in the next year and put some teeth into it. I'm just not sure that leaders get it. Well, just a little homework here. Deb is going to put some essays on the library website. They're related to the three talks that I'm giving, and some related materials. So you may or may not want to look at those. They're at least briefer summaries of stuff certainly that you heard last week and today. So just another comment here, and that is that oh, now are we stuck here again? Oh, there we are. One of the I mean, there are several things that you might ask, why am I doing this kind of thing? Why am I even interested in doing this Nobel series? Part of the original idea was just thinking about the relationship between the brain and the age of creativity. When is the best work, most novel work done by people? And it is interesting because if you, among people certainly like Albert Einstein and Niels Bohr and Paul Dirac at Cambridge, and for that matter, most theoretical physicists, there's no doubt that the most creative period in their career was in their 20s or early 30s. And that probably holds true for most mathematicians. I single those two areas out because they don't require a lot of, it doesn't take you kind of 10 or 20 years to get up to speed before you're judged kind of worthy or you're creating worthy work. And it also plays out with athleticism and age. I mean, just look at tennis and the three males that still dominated. But, there is an end at some point, and we've probably seen that with Roger and Roger Federer, and certainly any events that involve speed and dexterity. We're really talking about, that first 3 decades. Now, as someone pointed out last evening to me, well, Gordie Howe was still playing, I think, in his early 50s. Well, that was Gordie Howe. There aren't many other outliers like that. And I should say, I mean, we think about muscles and joints when we think about age and athleticism, but it's really about the brain, largely, about the brain being able to orchestrate all of this kind of motor activity on time. And one reminder of that to me recently was how quickly young people talk, but they also pick it up. And sometimes with people my age, I'm not as quick kind of hear what they're saying, or the rapid fire conversation from them. So they're processing stuff a lot quicker, at least than I am. I'm no expert in music, so I'm not going to say much, except most rock groups were young in their beginning. And yes, they kind of dragged it out over several decades, but perhaps most of their best work was when they were young. The visual arts is another thing, and literature, so I won't comment on those. Val may, Elkins or might say something about literature, but my sense about that is it takes time to mature. So anyway, that's part of it. The other thing is I have a great interest in human origins, and especially in that, well, what I think of as this threshold period to when we became what some people would say kind of fully human. Now that suggests an edge or a very sharp period, but it isn't. It probably happened over several 100,000 years. But those are the things that interest me, as well as in my career as a neuroscientist. That was a very focused laser-like career. But I did know several Nobel laureates. One was a mentor for me over almost 3 decades. And so I got to know some of them, and a lot of respect for it. Well, let's just look back at some of the, as I did last time, at more recent Nobel Prize winners in chemistry. And here are 3 that worked on the lithium-ion battery. And Stanley Whittingham, John Goodenough, isn't that quite a name, Goodenough? Akira Yoshino, and Goodenough is extraordinary. He's 97 now, and he's still productive. And Roger Penrose last year, 87, so he'd be 88 now, and still productive. So there are certainly exceptions. Just as a reminder, since we're talking about chemistry here, the hydrogen and helium and lithium that we talk about today and their related isotopes are the only elements that were created in the Big Bang. All the rest were created in stars and supernovae or collisions of neutron stars. So a difference in their origin. Now back to Goodenough. He's still actively involved in battery research. In fact, he's involved in a project with that combines Hydro-Quebec with Mercedes. That I guess they've divided up the cost 50-50. And the interesting thing is they are developing a solid state electrolyte, which incorporates a polymer to reduce the operating temperature of the battery from 80 to 50 degrees centigrade. Well, it almost doubles the range, and apparently their claim is that the charging time might be 5 minutes or less. That's a huge change, and we're talking about a range of maybe 1,000 miles, 1,600 kilometers. That's a lot, 3 times what some EV vehicles are getting now. So just moving along. Now there is a cost. I mean, it's very nice to drive e-vehicles, and I'll probably get one in the next year or so. But you know, industry estimates suggest that there might be 10 or 20 million of them on the road by 2025. But look at the cost. They involve a lot of cobalt and nickel. And the mining to get that stuff is kind of nasty mining on the environment and for the people doing it. And the global requirements by 2025 would be 200,000 tons of cobalt per year and 400 to 800,000 tons of nickel. So it's kind of a no-go there. And then of course, then you have to think about, well, where's the recharging energy coming from? If it's coming from coal or it's coming from a fossil fuel, what's been saved? And also then the issue of disposing of these batteries. That's a whole other challenge. Anyway, that's enough on that. Then moving to last year, 2020. So this was the year of CRISPR, gene editing. and Manuel Carpentier and Jennifer Doudna for the development of a method for genome editing. And I've watched that one for probably 5 or 6 years before then, maybe a little bit longer. There they are, the two of them. And so what better idea did those two have? Well, they learned from how bacteria fight viruses. Bacteria store memories, either the RNA if it turns out to be an RNA virus, like the COVID virus, or a DNA virus from previous encounters, and incorporates them within the bacteria's own DNA. So it has a template, a memory of previous encounters. When that bacterium encounters a new virus, it checks its catalog or library of previous encounters looking for a match. And if it finds a match, then it creates a guide, an RNA guide, to latch onto some part of the invading virus's RNA or DNA, and it has an attached protein enzyme that destroys the virus. So that's what bacteria do. That's what bacteria do in our gut to deal with some viruses and even some bacteria. And of course, it's not so different than our immune system. We have memory cells in our immune system, but the difference with us is that we combat viruses with antibodies and T cells, especially killer T cells. But still, the idea of establishing a memory and then panning through the most recent invader, looking for a match, that's very important. So now on to something it seemed quite appropriate this morning to bring this one up, because this is the Nobel Prize in Chemistry in 2018 to Francis Arnold and George Smith and Gregory Winter. Well, I'm not going to talk about George Smith or Gregory Winter, but I am going to talk about Francis Arnold because she not only has an extraordinarily interesting biography, which I'll talk about a little bit later, but she's clever, and she definitely had a better idea. So, and it is relevant today because we're talking about enzymes, things that break down molecules. In the 1980s, Arnold tried in vain to rationally design enzymes by changing the selection and order of amino acids in the protein molecule. But because the latter can be assembled in an almost infinite way, there are 20 amino acids that can be arranged in different orders. It was a very inefficient way to develop a designer protein, in this case, an enzyme. So she shifted to using what she called, her words, directed evolution, to modify an enzyme that she was working on at that time, subtilism, by creating random mutations in the enzyme's genetic code, and then introducing the mutated genes into bacteria, which then created different variants of the same enzyme, from which she selected the best performing variant, akin to natural selection, except that it was Francis that was doing the spelling. The spelling there, Francis, should be an E in there. Anyway, she repeated the sequence through enough generations to create the very best variant of this enzyme, and it could be hundreds of times more effective. So it was one of those really good ideas, and in today's laureates, one in particular, List, we'll hear about him later, had another really simple but brilliant idea. Well, here's the picture of that. Well, we don't need to do that because I said it in words. I do need to review a little bit of biology today, and I may even repeat myself a couple of times. That's no bad thing on the learning curve for most of us, but it is germane to today's subject. Humans possess about 25,000 protein encoding genes. That is, their job is to make one gene makes one protein. Actually, one gene can make 3 or 4 different separate or distinct proteins. But anyway, cells make around 100,000 proteins. Some of those proteins transcribe. So that's these are transcription proteins. That is, they activate or suppress genes, either single genes or whole families of related genes. Now, the obvious example starts with conception and with a single cell, fertilized ovum. And from that fertilized ovum, cells differentiate into thousands of types. I mean, I think the current count is up to 50 different neurons in the brain. There are probably even more subtypes than that. And so what's actually happening? I mean, all these cells have the same DNA, yet they look and behave very differently. So what's happened? Well, what's happened along the way is that whole suites of genes, hundreds, thousands of genes, have been silenced and others activated. That's what happens in normal development. Now, some genes in their product, their protein products, are very ancient. They date back to the earliest complex single cells. So we're going back 500 billion years plus ago. And we share many of those genes. They may not be exactly the same, right down to the last amino acid, or exactly the same base sequence, but very, very similar. Conservation of genes and their gene products over such long time periods strongly suggests that they were and continue to be vital to normal cellular function. Now, DNA does a really good job of copying itself without too many mistakes. It's certainly better than RNA, a lot better than RNA, but it makes mistakes. And most of those mistakes are neutral. They don't make any difference. Some, very few actually, confer an advantage. Some offer an advantage to some cells, but not others. For example, cancer. Others turn on a sequence of programmed cell death. I don't know whether you know this or not, but in early development, in the normal sequence, the brain probably develops 2 to 3 times the number of nerve cells that will be left. And the same is true in muscles, where there might be 2 times as many muscle fibers. And an active competition actually takes place for the best connections, the connections that are working. And for those who are disadvantaged or came second or third in the race, they kind of disappear without a trace, leaving no sign that they were there before. It's quite remarkable. There's no pathology left. They're just gone. Now, this is really ABC stuff. So there's the double-stranded DNA at the top here and the base sequence, tyrosine, alanine, cytosine, and so on along here, and then the matching one in the pair. So thymidine with alanine, thymidine and alanine, right? So once the 2 strands have separated, one of those strands is copied. You don't have to copy both strands, it's one strand. There's more to this than what I'm saying right now, but that creates the messenger RNA. Why is messenger such a good name? Well, it is a messenger. It's carrying the mail of the DNA code written in the base sequence from the nucleus into the cytoplasm and delivering it to where the proteins are actually made. So, here's the highly cartoonized ribosome in the cytoplasm, one of hundreds or even thousands of these ribosomes in the cell that are busily cranking out a protein here, amino acid by amino acid. There are 20 amino acids from which to choose. Here's the messenger RNA kind of going through here, and the code is read 3 bases at a time. So three bases code for 1 amino acid. All right, that's how it works. Now, something about protein function. What we're talking about is the one at the bottom of the list. Well, as I told you, there are 100,000 proteins, so there must be 100,000 functions or subfunctions, but we're only talking about the main ones here. Well, proteins, we may not be aware of this, but cells have skeletons. They have an interior skeleton, cytoskeleton, so to speak, that kind of prop up the membrane. And those skeletons consist of tubules with a core in them, and also solid filaments. And they can change the shape of the cell, push it out or retract part of the cell. They can also contract highly specialized fibers, such as muscle fibers. And they behave like a mail delivery system too. They transport molecules along. So it's a kind of molecular mail system. And they also are responsible during cell reproduction, and they made 2 copies. They act to kind of pull the chromosomes apart. I have already alluded to their role in activating and silencing genes. They play a role in the repair of DNA, the translation of messenger RNA, which I just showed you, into proteins. Next week is all about receptor proteins for sensation. So they play a big role with receptor proteins. They ferry molecules through the cell membrane. They provide the ion channel. And they're promoters for genes. And last, and highlighted in red, they function as catalysts. Now, just a little brief review here to kind of nail it down. So specific protein encoding genes code for specific proteins. That's what they do. It's very, very specific. So gene to messenger RNA, that's transcription. Translation is messenger RNA to protein. Proteins are chains of amino acids, of which there are 20. I've said that now. Each coded by 1 or more of these triplets of bases. The most important line, or 3 lines at the bottom, the function of a protein is dictated by the sequence and the types of amino acids in the chain. The 3-dimensional shape of the protein and the location of active sites. Now, if you've been following, and who hasn't these days, COVID and SARS-CoV-2, we're quite familiar with looking at pictures, cartoonized pictures and real pictures of the virus, but also kind of atom sized or molecule sized pictures of the spike protein. And those spike proteins contain several hundred amino acids in their chains. And the function of that spike protein is to make contact with the ACE2 receptor on our cells. And it's designed to do that really well, and it does do it really well. But some mutations might make it less likely to happen. Others facilitate that connection. So it would make the virus more transmissible, perhaps more lethal. And from what I've been watching, often there are 4 or 5 mutations at the same time, but the critical ones are the ones that have anything to do with the connection of the target cell. So that's very important to remember that conformational 3-dimensional shape of the protein, and therefore the exposure of certain charge groups is what's important for the function of the protein. That'll be reinforced later with what I say. And here, until this last year, mapping the 3-dimensional shape of proteins was really a laborious process. It took many hours and a lot of work. But now they have a library of several hundred proteins, and they've been used as teachers for artificial intelligence. And AI is now taking over, figuring out the molecular shape and structure of proteins and doing a really good job of it. And it's a lot quicker. So that's just happened in the last year. That's the reference, or one of the references at the top. Now, I've singled out one disease, Huntington's disease. I didn't see many cases of this, but maybe you're familiar with it. But it's a dominantly transmitted disease, neurological disease, and it causes it usually in midlife, sometimes a little earlier, behavioral changes, sometimes frank psychoses, but in the end, dementia, and associated with abnormal movements, kind of fidgety movements, so-called chorea. And what's it related to? Well, it's related to a mutant gene that creates long strings of the same amino acid. Now you think about that. It's like me sitting on the keyboard here, and instead of tapping out the next letter, A, let's say I'm making it up now, I keep my finger on the keyboard, I tap it again, and I get a long string of A's, hundreds of them maybe. That really distorts the protein, so it can't do its proper job. And in this case, sustaining nerve cells. And so it's responsible for the death of the nerve cells. The same thing happens in another disease. That's the fundamental molecular problem is the same, but it happens in myotonic dystrophy. I used to see a lot of people with this, and delightful patients, many of them that I remember, but the same problem. At one site in the protein molecule, there's a long stretch where it's the same amino acid again and again and again, and distorting the molecule, and therefore creating this kind of muscle problems for the patient. So structure is very important. Now we finally get around to this year, Nobel Prize in Chemistry. So we built a foundation here. And this is all about building better enzymes. Well, we heard about that a little earlier, using directed evolution. So this is another Nobel Prize given focusing on enzymes, and especially organic. Organic meaning life, meaning carbon-based protein enzymes. So Benjamin List and David W.C. McMillan, those are the two. And they're there. And those must be the same artist every year. Actually, these look pretty good. So building better catalysts, organic enzymes. Well, let's just talk about catalysts in this conversation. Catalyst and enzyme are the same. An enzyme is an organic catalyst, but not all catalysts are organic, might be metals, okay? So catalysts, what's peculiar about them? Well, they accelerate reactions without themselves changing. For example, a simple one. In fact, I was thinking about doing this here today. Put silver in a beaker, which contains hydrogen peroxide. So that's H2O2. The hydrogen peroxide changes into water and oxygen, but the silver is unaffected. So the enzyme, the catalyst, isn't changed, but the molecules have changed, right? Or in the case of our catalytic converter in a car, it controls the exhaust emissions that converts toxic gases and pollutants into exhaust gases from internal combustion engines into less toxic pollutants by catalyzing a redox reaction, meaning combined oxygen, O2, with carbon monoxide and other unburned hydrocarbons to produce water and carbon dioxide if it's working properly. Now, metals, just going to the bottom, are excellent catalysts because they borrow or share electrons willingly. By the way, that's an interesting property because in the life molecules, carbon occupies a unique position because it donates and it receives electrons well, but not so easily that that bonds can be broken. So bonds can be made with some facility, but they're durable bonds for the most part. So metals are excellent catalysts because they borrow and share those electrons willingly. But what's the downside? Well, the downside, many of them are heavy and toxic metals. So that's not it. Now, the other thing is that when reactions are being catalyzed, it often happens, especially when metals are involved, that you get two two mirror image molecules, same structure, but one's a right hand and one's a left hand. And those mirror images may have different functions. And in the case of this particular molecule, it tastes like an orange with one and a lemon the other, but can have serious side effects, and I'll mention that later. So let's get on with life's catalysts, or organic catalysts. So all living things have thousands of different enzymes. Organic ones, I've said this, I think 2 or 3 times, are carbon-based enzymes. Remember, I talked about CRISPR-Cas9 or CRISPR-Cas, the gene editing tool. Well, the Cas is a protein that's an enzyme. It could be 9, it could be 7 or 8 or a different number. Anyway, but that protein is an enzyme. So proteins are made of chains of amino acids. You've heard all of that. RNA, I didn't mention this. RNA also has enzymatic properties. Now, getting back to the evolution of life, RNA was probably the first, or something like RNA, was probably the first replicating, the first molecule capable of replicating itself, organic molecule capable of replicating itself. It's a single strand. It's somewhat unstable. It mutates more than DNA, but unlike DNA, it has enzymatic properties. So it has some pluses and minuses. By the way, that's our coronavirus, right? RNA. Organic enzymes are very efficient, and most do not incorporate a metal. Many specialize in asymmetric catalysis, meaning it only creates the one hand, not both, not 2 mirror images. They also, and this is a great, talk about social cooperation. They also work side by side. When one enzyme is finished with its reaction, another takes over in a chain. And in this way, they can build quite complicated molecules or be part of a standard metabolic chain, such as oxidative phosphorylation in mitochondria, that provide almost all of our high energy phosphate, and the Krebs cycle. So it's like racers passing the baton on to the next one in a relay. Well, Benjamin Liss, what was his better idea? I mean, I mentioned earlier that it would be simple, and it was incredibly simple, and he was stunned that it actually worked. I've mentioned earlier that proteins are often involve hundreds, if not thousands of amino acids. And most naturally occurring organic enzymes, they're all proteins, are huge. But the odd thing about it is the reactions those large molecules catalyze are often driven by only one or few of the amino acids in that string of hundreds or thousands. So Liss asked the obvious question: do you really need that whole stretch of hundreds of amino acids or thousands? Or can he get by with just one of the amino acids, or maybe two or three? Well, why not indeed? When he thought about this, he went into the literature and he couldn't find anything on this. He thought, oh, well, I'm sure that people have tried this out. This is silly. This is so obvious. Somebody must have caught on to this one. Well, they hadn't. And he showed that the first one, the amino acid proline, which is a simple one and cheap and environmentally friendly, was capable of catalyzing organic molecules. So, wow, that was talking about a game changer. And here's an example of a very complex, one of these, it's not reproduced well here, but very complex a protein molecule. And only in one place in here is found the kind of active amino acid that's actually doing the job, or the active groups. So, so that's an example of that. Now, what about David's ideas, achievements? Well, this a little, it was less obvious to me, so it'll probably be less obvious to all of us. Organic molecules are the building blocks of all life and contain simple and common elements, such as stable framework of carbon atoms, as well as atoms such as oxygen, nitrogen, phosphorus, and sulfur. By the way, I've just listed off the molecules of life, or at least the atoms of life, right? So, so carbon, oxygen, nitrogen, phosphorus, sulfur, it's probably the same physics and anywhere in the universe where life might have appeared, has appeared, or will appear. I mean, it's going to, the same rules will apply. So he began to design simple organic molecules, which, like metals, could accommodate electrons. It's all about sharing, donating, and receiving electrons, which are far more stable, as it turned out, than metals, without the downside of metals. To catalyze the reaction he was interested in, it needed to form ammonium ion, which contains a nitrogen atom that has an inherent affinity for electrons. He selected several organic molecules with the right properties and tested their ability to drive a common reaction that chemists use to build rings of carbon. Well, guess what? It worked. It worked brilliantly. And some of the molecules were excellent at asymmetric catalysis, meaning just one hand. No confusing two hands. Well, what are the applications on this field? Well, the development and manufacture of antihypertensive drugs that are really in the process of revolutionizing that. The development and manufacture of monoclonal antibodies for the treatment of cancer, that's been around for at least 10, 15 years. Autoimmune diseases, rheumatoid arthritis, you name it, there's a great list of them, about 100 of them now. Monoclonal antibodies, and acute viral disease. Well, we saw that President Trump got a kind of a monoclonal antibody treatment. Remember that? Well, the rest is history. Benjamin List and David McMillan discovered an entirely new concept, organocatalysis. They developed many cheap and stable organocatalysts to drive a huge variety of chemical reactions. They're far more efficient and forgiving to work with, and they avoid that symmetric catalysis that occurred with thalidomide. Remember thalidomide back in the 60s and the deformities of babies? That was caused by one image, one of the two mirror images of thalidomide. So, well, where are we in biology these days? I mean, it's interesting, the kind of string of triumphs. I look back, I see the Nobel Prize, they in no way represent more than probably 0.1% or less than the stuff that's happening in science these days. There's a lot going on, and some of the stuff that's somewhat disturbing to me and to others is the fiddling that's going on with the molecules of life. So it started, as far as I was aware of it, maybe 5 years ago, with introducing artificial bases. So instead of accepting the 4 bases provided by nature, they substituted an artificial base. And then they went from 1 to 2 to 3 to 4. So they've just changed the whole DNA molecule. So they made entirely new DNA. That made possible making new proteins because if you change the gene, then you change the protein. Another spelling error on Francis Arnold there. So, and why not entirely new life forms? Well, actually, that's been tried. And it is possible to create new life forms, meaning a self-replicating molecular structure. I mean, we're not talking about complexity here, but it does bring up this whole question of weaponizing viruses or bacteria by changing existing forms or even creating brand new ones. So that's the worry. Now, this is David McMillan, he looks quite happy, doesn't he? I envisage his next office will be larger. We'll see. Now, I thought you might be interested in this one. This is the process for selecting who wins the Nobel Prize. And this slide has come from that because the process begins 16 months. I mean, it takes 16 months. So in September, in a cycle, just follow this through. The nomination invitations for nominations are sent out in September. Now, this is for the science ones. I don't know about the other ones. Were sent out in September with a deadline for receipt of the last one on the 31st of January. Then there's a period of evaluation. It goes on beginning through March, April, and May. I would think there's a lot of work going on in here of reading all of these things and trying to make sense of them and trying to put them in some kind of hierarchy or order. If you're not sure yourself, sending it out to others. I think the Nobel Committee outsources a lot of this stuff. There are the obvious people, previous Nobel Prize winners, but they, at least in the writing, any professor of a reasonable university around the world is included, can submit nominations. I rather like that. And now the time's getting short here because now they've got to go, am I probably reducing something like 100 or 50 or 30 down to who gets it here. It's a majority vote. And boy, they leave that vote fairly late in the process. I mean, it's in September when that vote is taking place. And then they announce these prizes. I think they started on October the 5th this year. And so that's the process. There's a lot more that can be said about this, and I'll add some more in another talk, just so this is a bite-sized piece. Now, in preparation for next week, you actually have some homework because we're talking about sensation next weth, and sensations of touch discrimination, and thermal sensation, warm, cool, and when it becomes hot and freezing, and when it becomes painful. So pain, touch, and thermal sensation, and another one, muscle sense. Our sense of the rate of change of muscles as they lengthen or contract to provide some sense of joint position. So one thing you might try out on yourself is to, one simple thing, I don't know whether you can see this, but if you take your thumb, just for a joint position sense, and you can work with a partner or a friend, and just as I've done thousands and thousands of times with patients, you say, I'm going to move your thumb up or down, up or down. All right, now keep your eyes open and look at it and say, tell me what I'm doing, up or down. And then, so we're on the same page. Now close your eyes and tell me whether I'm moving it up or down. And you can use large angles here, 10, 15, 30 degrees. Try to see what the minimum is. What is the minimum? Okay, some of that information is coming from muscles in the hand and in the forearm, and some of it's coming from the skin. That's where that information is coming from. So just try that out. The other thing, which I made up here, and then I couldn't find this morning, but you can take let me see if no, I don't have it. Anyway, if you've got some wire at home that you can kind of bend, so you've got 2 points in the end and kind of millimeter or half millimeter points in the end, so that you can change the distance between those 2 points, and then try them out on your finger, and just see how small a distance between those 2 points can you actually resolve on your fingertip, and then move back in your finger. Remember the homunculus? We did the brain series, and that disproportionate area, the cortex that was committed to the lips, here, and the eyes, and the tongue, and to the hand, and especially the ends of the fingers and the thumb. Okay, well, that's reflected in the sensory. So you can actually work that out, what that would, and try it out over the back of the hand, or the shoulder region, or on your tummy, and just see, can you actually resolve 2 points only 3 or 4 millimeters apart on your chest? I can tell you, you can't, but, or anything like it. So just try that out on yourselves. It gives you a feel for next week's talk on sensory receptors. Oh, by the way, these sensory receptors, that's a whole world there that's changed so much just in the last few years, because now people were asking very fundamental questions. Okay, what actually transduces that touch into an electrical signal that's sent up to our brain? But what's actually translating that stretch or touch or thermal sense into that electrical signal? So that protein, that receptor, those receptors are what we're going to talk about next week. So there may be questions from last week or comments, I don't know, but you can always email me. But if you have a question now, that would be fine.